Neutrophils make up the first wave of cells that cross the blood vessel wall to enter inflammatory sites. Here we give a step-by-step account of how the required effector cells are recruited into sites of infection. Under normal conditions, leukocytes are restricted to the center of small blood vessels, where the flow is fastest. In inflammatory sites, where the vessels are dilated, the slower blood flow allows the leukocytes to move out of the center of the blood vessel and interact with the vascular endothelium. Even in the absence of infection, monocytes migrate continuously into the tissues where they differentiate into macrophages. During an inflammatory response, the induction of adhesion molecules on the endothelial cells, as well as induced changes in the adhesion molecules expressed on leukocytes, recruit large numbers of circulating leukocytes, initially neutrophils and later monocytes, into the site of an infection. The migration of leukocytes out of blood vessels, a process known as extravasation, is thought to occur in 4 steps. The first step involves selectins. P-selectin, which is carried inside endothelial cells in Weibel-Palade bodies, appears on endothelial cell surfaces within a few minutes of exposure to leukotriene B4, the complement fragment C5a, or histamine, which is released from mast cells in response to C5a. The appearance of P-selectin can also be induced by exposure to TNF-alpha or LPS, and both of these have the additional effect of inducing the synthesis of a second selectin, E-selectin, which appears on the endothelial cell surface a few hours later. These selectins recognize the sulfated sialo-Lewis moiety of certain leukocyte glycoproteins that are exposed on the tips of microvilli. The interaction of P-selectin and E-selectin with these glycoproteins allows monocytes and neutrophils to adhere reversibly to the vessel wall, so that circulating leukocytes can be seen to roll along endothelium that has been treated with inflammatory cytokines. This adhesive interaction permits the stronger interactions of the next step in leukocyte migration. This second step depends upon interactions between the leukocyte integrins, LFA1 and MAC1, with molecules on endothelium such as ICAM1, which is also induced on endothelial cells by TNF-alpha. LFA1 and MAC1 normally adhere only weakly, but interleukin-8 or other chemokines bound to proteoglycans on the surface of endothelial cells trigger a conformational change in LFA1 and MAC1 on the rolling leukocyte, which greatly increases its adhesive properties. In consequence, the leukocyte attaches firmly to the endothelium and rolling is arrested. In the third step, the leukocyte extravasates or crosses the endothelial wall. This step also involves LFA1 and MAC1, as well as a further adhesive interaction involving an immunoglobulin-related molecule called PECAM, or CD31, which is expressed both on the leukocyte and at the intercellular junctions of the endothelial cells. These interactions enable the phagocyte to squeeze between the endothelial cells. It then penetrates the basement membrane, an extracellular matrix structure, with the aid of proteolytic enzymes that break down the proteins of the basement membrane. The movement through the vessel wall is known as diapedesis and enables phagocytes to enter the subepithelial tissues. The fourth and final step in extravasation is the migration of leukocytes through the tissues under the influence of chemokines. Chemokines, such as interleukin-8, are produced at the site of infection and bind to proteoglycans in the extracellular matrix. They form a matrix-associated concentration gradient along which the leukocyte can migrate to the focus of infection. Interleukin-8 is released by the macrophages that first encounter pathogens and recruits neutrophils, which enter the infected tissue in large numbers in the early part of the induced response. Their influx usually peaks within the first 6 hours of an inflammatory response, while monocytes can be recruited later through the action of the chemokines such as MCP1. Once in an inflammatory site, neutrophils are able to eliminate many pathogens by phagocytosis. They act as phagocytic effectors in an innate immune response through receptors for complement and other opsonizing proteins of the innate immune system, as well as by recognizing pathogens directly. In addition, they act as phagocytic effectors in humoral adaptive immunity. The importance of neutrophils is dramatically illustrated by diseases or treatments that severely reduce neutrophil numbers. Such patients are said to have neutropenia and are very susceptible to infection with numerous pathogens. Restoring neutrophil levels in such patients by transfusion of neutrophil-rich blood fractions or by stimulating their production with specific growth factors largely corrects this susceptibility. And that's neutrophils make up the first wave of cells that cross blood vessel wall to enter inflammatory sites. Have a great day.